Awesome. It is now 6:30. We are going to get started with today's webinar. If you have any friends or family you know who aren't logged in yet, tell them not to worry at all. This webinar will be recorded and we'll be posting it to our YouTube channel tomorrow. So welcome everybody to the LTVCA's third annual winter webinar series. The last 2 years have been such a success that we brought it back for a third year, and I'm very pleased to enter our first webinar of the year here, Ecotoxicology, Contaminants in Your Environment, with Ph.D. students Quinn, Allamby and Collen, Wardlaw. If you don't know what ecotoxicology is, do not worry, you are going to learn all about it today. But first, I'm just going to give you a quick intro about the Lower Thames Valley Conservation Authority and what we offer all of the residents of our watershed. So before we begin, I'm just going to do a First Nations acknowledgement. Yep. Some technical difficulties here. As I do this First Nations acknowledgement though, please keep in mind that we're really encouraging people to go beyond just saying these words, just acknowledging these things, and really thinking about what they mean and how you can internalize that to take action yourself. So we will begin by acknowledging that the land on which we gather is the traditional territory of First Nations people who have long-standing relationships to the land, water, and region of southwestern Ontario. We also acknowledge the local Lower Thames River watershed communities of this area, which include Chippewas of the Thames First Nation, Oneida Nation of the Thames, Montee Delaware Nation, and Caldwell Nation. We value the significant historical and contemporary contributions of local and regional First Nations and all of the original peoples of Turtle Island. We are thankful for the opportunity to live, learn, and share with mutual respect and appreciation.
Just before we go on, Alyssa, there is a slight echo, I think, happening from your mic. Not sure. We can hear you. There's just a slight echo coming back.
For sure. Let's try this. Awesome. How's that?
That's better.
Okay. So there we go. Awesome. So to those of you tuning in from our watershed, I'm sure you're familiar with the Lower Thames Valley Conservation Authority. For those of you that aren't, we are actually one of 36 conservation authorities in Ontario that all work together to protect and enhance the environment. So we do that through a few different ways. We have whole departments that are dedicated to water quality, species at risk, stewardship, planning and regulations, and agricultural programming. And we all work together to offer services to our residents and make sure that we're being safe and interacting respectfully with our environment. Now, we do offer services to those that live in the watershed. So as you saw in the image there, we extend all the way in southwestern Ontario from Windsor up towards London. And we have funding available for drainage buffer strips, windbreaks, prairie, tree planting, wetland, and farm field cover crops. If you have land that you think that you would like to do a project with us, we offer a lot of funding and obviously technical support. So please feel free to email stewardship@ltvca.ca. Now, one of the sub-programs that we have at the LTVCA is the Urban Stewardship Program, which focuses on engaging the public in restorative activities throughout the watershed. So for those of you that don't have huge swaths of land to restore, but you still want to be involved, you can always volunteer with the Conservation Authority for all of the things listed on this slide. So things like tree planting, litter pickups, removing invasive plants. If you're interested at all in volunteering or you have a group that you would want to do a dedicated event with, please feel free to email the email on the right side there, urbanstewardship@ltvca.ca. Now, just some upcoming events that we have going on at the authority. Our big one right now is our tree seedling order form. Again, if you're looking to plant a project with us or simply just buy some seedlings, our tree order deadline is on March 1st, which you can find on our LTVCA website or our social media, or just by coming into our office right here in Chatham. We also have the CK Farm Show that we will be having an outreach booth at tomorrow and Wednesday, where we're going to be talking about all of our programs and everything that we offer to farmers. So make sure to check that out there. And of course, this is only the first installment of the winter webinar series, and we have 3 more, the second of which is live for sign up. Again, you can find that on our social media or our website. Now, just a quick introduction for our presenters today. As I said, Quinn and Colleen are both Ph.D. students at McMaster University, specializing in ecotoxicology. So Quinn's Ph.D. research aims to assess the toxicity and accumulation of a variety of microplastic sizes and polymer types on different freshwater benthic macroinvertebrates using environmentally relevant exposures. Additionally, her research investigates the impact of microplastics on the microbiome, their potential for bioaccumulation, and transgenerational toxicity. Quinn's research is a part of a larger project investigating the effect of microplastics on biota in Canada's freshwater and soil ecosystems. Now, Colleen is currently working on her PhD as well, but looking at the transfer of microplastics from aquatic to terrestrial food webs through emergent insects into nearshore spiders. This work takes place in the Grand River watershed near wastewater treatment facilities and is part of a larger investigation characterizing the exposure, effects, and risk of microplastics to freshwater and terrestrial ecosystems in Canada. Now, again, if any of those words went right over your head, don't you worry, you are going to get a very in-depth look into what ecotoxicology is, and hopefully all of that will make a lot more sense to you. Okay, now I'm going to hand it over to our awesome presenters. I am going to ask if you have any questions, please do not put them in the chat because they might get lost, but put them in the Q&A tab. We'll be having a Q&A period at the end. So again, if you have any questions, feel free to enter them there and we will ask our hosts about that at the end. All right, we hope you enjoy.
All right, I'm just gonna start sharing. Which one? Can you see our screen?
I can see it.
Awesome. Great. Well, thank you so much for the introduction. We're super excited for the Lower Thames Valley Conservation Authority that they've organized this opportunity for us to give you guys kind of an inside look at what ecotoxicology is and how it applies to your environment. So my name's Quinn, this is Colleen, and we're super happy to kind of go over this and get into it.
So as I'm sure we are all aware, pollution is everywhere. And whether this is accidental oil spills, mercury in our fish, or the air we breathe, our world is troubled by environmental pollution. And unfortunately, the vast majority of this pollution is related to human activity. So what we hope to go over with you guys today is how do we know which contaminants are a concern and how can we test this? To understand how the pollution might be affecting our environment, we have ecotoxicology. Eco refers to the ecology or the environment, and toxicology is concerned with the nature, effects, and detection of hazardous chemicals. So putting this together, ecotoxicology is looking at how contaminants can harm our ecosystems, both aquatic and terrestrial. Ecotoxicology looks at adverse effects of various contaminants on living organisms and their interactions with the environment, where adverse effects means harmful or abnormal result or reaction from the exposure of the contaminant. So an example of this might be a duck caught in an oil spill will have oil matted to its feathers, and the adverse effects caused by the oil coating will impair its ability to fly, which can exhaust the animal as well as reduce the ability of the duck to regulate its temperature. So negative adverse effects.
So what is a contaminant and why should we care to study contaminants? So contaminants are substances that are accidentally or deliberately enter the environment. They are not often, but not always, because of human activities. Once these contaminants enter the environment, they have the potential to cause harm to the ecosystem that they're introduced to and the organisms that live within that area. They also have the potential to enter the food chain where they may accumulate.
So once a contaminant has been identified, how do we determine the risk that a contaminant may pose to the environment? The main goal of ecotoxicology is for us to try to understand what the level of contamination already is present in the environment, At what concentrations does the contaminant pose a risk to the environment or its living things? And finally, what this means on the broader scale for our environment.
So the main goal of toxicity testing in the lab is to determine if a specific contaminant is toxic to specific organisms, and if so, at what concentrations do we start to see lethal and or sublethal effects. And effects in this sense are usually measured in terms of overall survival, which are lethal effects, or growth and reproduction, or sublethal effects. But they can also be measured in terms of behavior and or other attributes. These tests usually follow specific or standardized methods or instructions that are created by different organizations. These standardized methods are used to help reduce the variability within these tests. So when we're determining the toxicity of a contaminant, so that even if a different lab or organization runs a toxicity test on a specific contaminant, the outcomes should be the same. And these standardized methods include both soil and water tests using specific organisms. So for example, if we wanted to determine the toxicity of a contaminant in soil, we could employ standardized methods that have already been created using earthworms. If we wanted to see how a contaminant might impact aquatic environments, Daphnia or water fleas are a very common test organism used in standardized testing. Rainbow trout are another common aquatic organism that also has standardized methods.
We can also use field surveys to determine if a contaminant is present in the environment and also in what concentrations. We use field surveys to monitor the status or impacts of a contaminant. If a contaminant once was detected in a particular location, we continue to visit the site to check if levels seem to be increasing or decreasing, but we can also look for changes in the environment in response to the contaminant present. We also like to use field surveys to check the status of organisms to see if they've been impacted by the contaminant and then monitor the recovery. Now together, our lab toxicity testing that Quinn went over and our field surveys help to inform on risk assessment. And risk assessment is going to help us to understand the hazard, what the contaminant present is and in what concentrations, the receptor, which is who is being impacted. It might be humans, it could be animals, maybe a particularly sensitive species in our rivers. And then we also consider the pathway, which is the how the contaminant is moving in the environment and coming into contact with our receptors. So an example of this would be a broken pipe would be the pathway, the leaking oil would be the hazard, and the exposed fish would be that receptor. Now, the concept of risk is at the intersection of these elements, and risk is defined by the likelihood of a contaminant exposure occurring and the harms that are caused by that exposure if it does occur. And we'll use these risk assessments to predict the likelihood of future events, evaluate the likelihood that the observed effects are caused by past or present ongoing exposure to the contaminant. And then we also like to use risk assessment to advise policy of what we can do and how we can try to change it.
And contaminants can have a wide range of effects that have the potential to span across many different organizational levels of an ecosystem. So we can see effects at individual levels, which are impacts to one organism in specific. So for example, this could be impaired reproduction or growth within an individual fish. Population level effects include effects that can be seen across an entire population of the same species. So an example of a population level effect could be that the reduced reproduction of that individual level is causing reductions in the population size. Community level effects include things like predator-prey dynamics or interactions between different populations of organisms in a specific area. So for example, a contaminant may eliminate a species essential to the functioning of the entire community, which may promote the dominance of another species, or it may simply decrease the numbers and variety of species present within that community. And finally, ecosystem level effects, which consist of different communities of organisms associated within a physically defined space. For example, loss of fish production in a stream due to the death of invertebrates from a contaminant. And contaminants that enter the environment also have the potential to bioaccumulate or biomagnify in food webs. So bioaccumulation, this is a common term that we hear a lot. It's regarding one singular organism, in this case, a fish, and it's the gradual buildup of contaminants in living organisms at a faster rate than can be excreted or broken down by that organism. They can enter and accumulate within an organism through exposures to contaminated air, soil, waste, or water, or food. So in the case of our fish here, over time, contaminants like mercury, for example, can start to accumulate or build up within that fish, leading to higher and higher levels of the contaminant. Biomagnification is also a pretty common concept. It's pretty similar, except that it considers how a contaminant may move up or biomagnify up the food chain. So this is generally through predator-prey interactions. So for example, in the case of our singular fish, when a larger predator like a seal, for example, consumes that fish plus other fish that may have been accumulating mercury, we can see that the concentrations of mercury increases as we move up the food chain.
So to this point, we've given you some background on ecotoxicological concepts, and now we're going to switch gears to give some real-world examples of how common contaminants we see across the planet. So we use pesticides globally for a number of things, like in agriculture to help protect our crops. And we also have personal uses for pesticides like bug spray, ant poison, rat poison, or even the flea and tick medications we give to our pets. And the main goal for our pesticide use is to control, deter, or kill unwanted organisms. And this can range from bugs using insecticides, plants, herbicides, fungicides, and rodents, rodicides. But the use of pesticides, while very beneficial to our society, also can come with some consequences, such as pesticide applications for agriculture may rinse into our waterways, and the pesticide residues in surface water can harm plants, animals, or contaminate groundwater. Pesticides may also reach non-target organisms, such as a mouse that has ingested poison may be hunted by a bird of prey like an owl, which will then poison the bird. And even if we overuse pesticides, we can render them ineffective against pests and they become resistant, which means less options for pest management or improved pests.
Another example of a common contaminant would be oil. So more than 4 million barrels of oil are moved through Canada every day, mostly transported by cargo ships, rail, transport trucks, and pipelines. However, at any point in the oil extraction, transportation, or storage process, we can see oil spills. Once it's released into the environment, oil can pose a huge risk to the environment. Oil in high enough concentrations can poison animals through internal or external routes of exposure. So for example, recent toxicity studies have shown that even small amounts of petroleum hydrocarbons can impair the successful development of fish eggs and embryos. Oil can also pose a physical risk to organisms like the duck with the oil. Birds and mammals often die because oil fouls fur and feathers and that they can no longer insulate themselves. Smaller organisms can also be smothered by thick layers of oil washing ashore.
And something a little bit more seasonal, something some people may not consider to be a contaminant is road salt, and it can actually pose some serious issues for the environment. We commonly use road salt to help manage our snow and ice conditions on roads and walkways in the winter, and it's really effective as long as our temperature isn't too low. But once we overuse our road salt, because of our abundance of caution to slips and falls, sliding, we tend to overuse it. So in Canada, we apply roughly 5 million tons of road salts every year. But overusing road salts will cause some issues. They get rinsed into our waterways, and once it dissolves, it will increase chloride levels of our waters, meaning our freshwater rivers are turning much more salty. And the increased salt conditions can be harmful to organisms like fish, amphibians, mussels, and bugs, as well as our aquatic plants. Some studies have found disruptions of reproduction, growth, and movement of organisms once the chloride conditions in our water increase. And some people have suggested that beet juice could be a good alternative to road salt because it does help melt our ice. But recent studies are actually cautioning against beet juice because it's more toxic to organisms due to higher potassium levels that will disrupt the organisms. So some alternatives we should consider. Homeowners can use standard kitty litter to help give more traction on snow or ice, or try to keep in mind appropriate salt application, which is approximately one standard coffee mug full of salt for a 2-car driveway.
There's also a growing concern about the occurrence of pharmaceutical contamination in our environment, with many of the more than 4,000 prescription medications ultimately finding their way into our environment. Pharmaceuticals can get into our water supply via human excretion and drugs being flushed down the toilet, but they can also be found in high amounts in wastewater from pharmaceutical manufacturing. While wastewater treatment plants typically help to treat wastewater from residential and industrial uses, pharmaceuticals are often still present in treated wastewaters that are released into aquatic environments. This suite of contaminants can have many adverse effects when introduced to the environment. For example, they have the potential to affect both the health and behavior of wildlife if they're exposed. So for example, perch that feed on damselfly nymphs that were contaminated with an anti-anxiety medication became bolder and left their schools more often. They also have the potential for both bioaccumulation and biomagnification, which we learned a little bit earlier in our presentation, with predators found to have higher levels of pharmaceuticals than what's actually present within the environment. Finally, arguably one of the most well-known environmental contaminants of concern right now is plastic pollution. We use plastics virtually everywhere. However, Canadians alone generate 3 million tons of plastic waste each year. This waste can often end up within our environment. And plastic pollution is concerning because they can take a very, very long time to break down and tend to accumulate in oceans, lakes, rivers, and shorelines across the globe. If organisms are coming into contact with these plastics, they can become tangled or they can consume them, which is very concerning. So both Colleen and I researched the impact of plastic pollution to the environment. So from here, we'll be providing an overview of what we do as ecotoxicologists and relating that to some of the concepts that we've learned.
Okay, so Quinn and I both kind of specialize on microplastics. So this stems from our plastic pollution issue. Microplastics are plastic particles that are less than 5 millimeters in size, and they're found very problematic due to their small size, high mobility, and ever-increasing abundances in the environment. Microplastics are a pretty diverse group of contaminants, and they are found both in aquatic and terrestrial locations, as well as the Arctic, virtually everywhere. And they are contaminants of concern due to their wide array of sources. Microplastics can be made in their small size, which is primary production, or they can be produced from the breakdown of larger items, which is secondary production. And secondary tends to be a little bit more common in what we see in our environmental samples. We tend to categorize microplastics based on their shapes shown here, which are fibers, fragments, foams, films, pellets, and beads. And these shapes can sometimes be linked to their usage or how they are produced. Now, within microplastics, we have a number of concerns, such as their small size, which allows them to be transported over very long distances, their accessibility to organisms, and their ability to leach chemicals that are in them with their production, as well as absorb environmental contaminants like pesticides or mercury have been found to stick to microplastics. And with the many problematic attributes of microplastics, our goal is to understand their occurrence, fate, and consequences once in the environment.
And just to give some quick examples of where microplastics can come from, we typically see beads from personal care products. We see fragments from the breakdown of larger plastics such as water bottles. And most commonly, we see fibers from the laundering of our synthetic fiber clothing items. So every time we're washing our synthetic fibers, they're releasing tons and tons of microfibers into our wastewaters where they eventually get treated, but most of the time, some still get through and are released into our waterways. So within freshwater systems, we've identified 4 key sources of microplastics, including agricultural runoff, wastewater effluent, industrial effluent, and stormwater runoff, which are all capable of releasing large amounts of microplastics into our rivers and lakes.
So on the topic of microplastics in rivers, we thought you guys might be interested in a case study which is actually conducted within the Lower Thames River, as well as the Upper Thames. So as researchers, we are interested in looking at microplastic pollution in rivers, specifically because they can transport microplastics in their flowing waters into larger water bodies, such as lakes and oceans. But rivers also can hold on to microplastics and accumulate them in the sediment. So the study I'm going to go over very quickly was done at University of Western Ontario in London, and researchers went into the Thames River at 34 locations shown here and collected samples of sediment from the river, which is the muddy or rocky bottom. So they looked at these sediment samples to see what kind of microplastics had settled in different areas and they found between 6 to nearly 2,500 microplastics were present per kilogram of sediment, which is pretty high. So the areas with higher microplastics tended to be samples that were being taken from city areas like Chatham, Woodstock, or Mitchell. But the sites located in London had the highest amounts of microplastics. And the most contaminated site was downtown London, where the two branches of the Thames River meet. And the study also learned the characteristics of river sediment matter. So the river if it's straight or bending, can accumulate plastics differently. And also if the sediment tended to be made of large rocks compared to smaller sand or mud. And these elements can be related to how our river is holding on to this plastic pollution. And related to the sediment study, while I was at Western, I worked on a fish part of this study. And because we knew the number of microplastics in the sediment, we then wanted to check if the fish that have feeding behaviors closely associated with feeding in the sediment along the bottom of the river might be ingesting microplastics. So here we looked at white sucker and common carp from 11 locations, but just from the upper Thames region of the Thames River. So with this study, we found that these bottom feeding fish had microplastic fragments, fibers, and even contained tire wear particles, which are generated from your car tires wearing on the road and then washing into the river. So we had found that 44% of the white suckers that we had collected and 31% of the common carp we collected contained at least one microplastic item in its stomach. So this shows us that not all fish had microplastics. But we did find that fish collected near city areas contained higher amounts of microplastics than fish collected from the farm areas. And this was similar to the sediment study. We also saw that the fish collected from London contained more microplastics. So overall, we found that monitoring the amount of microplastics in the sediment could be a good tool to predict maybe the amounts of microplastics that these bottom feeding fish are coming into contact with and ingesting.
So more on the toxicity side of the ecotoxicology, we're currently trying to determine the ecotoxicological effects that microplastics may have on freshwater macroinvertebrates. So these organisms, which include things like snails, mayflies, aquatic worms, are a very important component of freshwater ecosystems and are a super important prey item for larger organisms like fish. So our goal was to conduct toxicity tests using these organisms, using concentrations and types of microplastics that we've seen in the environment. And so far, we've seen that macroinvertebrates are tolerant of microplastics at a range of environmentally relevant concentrations and types. So they're tolerant of microplastics at concentrations that are typically found in freshwater rivers and lakes. But we've also found that they're capable of ingesting large amounts of microplastics. So as you can see on the photo on the left, that is species leaving the body of a mayfly larva. And all of that red fibrous stuff, that's all microplastics leaving the body of that organism. In the photos on the right, those are the fecal casts of snails. And you can see that they're absolutely filled with the same type of plastic, which are those microfibers. And we created these microfibers using a red polyester blanket that we got from Amazon, and we really only had to use a tiny, tiny amount of that blanket. And it's created so many microplastics, and these organisms are ingesting lots of them. However, we're still not sure if the microplastics have the potential to bioaccumulate in these organisms. So that's our next step of the project.
So on the other side of this, we are also not very sure at the moment about the presence and fate of microplastics in the environment. So here, my PhD research is aiming to investigate microplastics being sourced from wastewater treatment facilities, as Quinn mentioned earlier, are a source of microplastics to freshwater rivers, and also the movement of microplastics in food webs under natural settings. So to do this, we are looking at the amounts of microplastics that are coming from the wastewater treatment facilities and entering the water via effluent, which is the water being discharged from the facility into the river once it's cleaned. And we are also looking at the number of microplastics that are present in base food web resources. So this would be the biofilm, which is the green algae stuff that is growing on our rocks that make the rivers slippery, as well as the number of microplastics present in the sediment. We're also aiming to look at aquatic insects to see how much microplastics they're ingesting under natural field conditions, and considering if predators such as shoreline spiders are also being exposed to the microplastics, but through their prey when they eat the bugs. So together, the data I'm gathering in the field, as well as the data that Quinn is getting in the lab for toxicity, Quinn and I are hoping to inform on the concerns related to microplastics in freshwater ecosystems.
And while we're still trying to understand the impacts of global plastic pollution, and sometimes it can seem like there's only doom and gloom, there have been many global efforts to help mitigate plastic pollution. So some of these things include the ban of microbeads. So we saw that they were typically in personal care products. So those were banned a little while ago. Reductions in the use of single-use plastics. I'm sure we've all seen that and felt that at home. Development of legislations that hold companies more accountable for their plastic pollution, ocean cleanups, and improved general awareness. And this isn't just on the plastic front, that's also on many different contaminants across the globe. So while it may seem like it's lots of doom and gloom, there's lots of good work going on across the globe. And so finally, we also want to touch on the many ways in which we can make individual efforts to help our planet, especially with specific contaminants and what we deal with in our everyday life. So a big one is the reduce, reuse, repair, and recycle, including buying fewer things, shopping secondhand, and repairing what we can.
Another effort we can make is to advocate by speaking up again about environmental issues and encourage others to get involved, as well as voting for representatives that support your environmental beliefs and writing to MPs about environmental issues and why they matter to you.
We can also be more responsible consumers, making sure we're aware of the environmental impact of our purchases, making sure that we're aware of how to properly dispose of our waste, and supporting local businesses and locally made products.
And finally, we can all use a little cleanup, so try to get involved with your local cleanup events at your parks, rivers, beaches, and beyond. And to end off, we'd just like to thank the Lower Thames Conservation Authority for inviting us to give you an overview of what ecotoxicology is, as well as what we're trying to accomplish with our research. And should anyone like to contact us for any reason, our information is provided here. Thank you.
Thank you.
Thank you. That was very informative. I just want to say that I personally learned a lot. I loved seeing at the end everything that we could do. And just a reminder to everybody out there, if you know somebody that would be interested in any of this information, we did record this and this will be available usually on our YouTube channel. But if at any time you want to call us or give us an email, we can send it to you directly. And then we can also now we can open it for the Q&A session. I'm not sure, Alyssa, if there's anything you wanted to add before I did that.
Yeah, just again want to plug our Lower Thames Valley Conservation Authority stewardship programs. If you have any land that you're thinking of retiring, are tired of farming, or are having some drainage issues, please feel free to contact us at stewardship@ltvca.ca or just go onto our website. Again, we have lots of funding and expertise to give you. And again, if you liked this webinar or like webinars in general, please sign up for the second installment, The Art of Nature, which will go into all about the art of being a naturalist and how to kickstart your naturalist journey.
Awesome. So I think we'll start now with some of the Q&As here. And don't worry, I'll try to get to everybody's, so you can keep sending in your questions and we'll make sure we don't miss anybody, okay? So I'll start with question number 1 from S. Tweedle. The slideshow indicated that agricultural/agri practices were on the top 4 sources of releasing microplastics into the environment. Can you expand?
Yeah, so I can touch on this a little bit more. So in the lab right now, we're currently working on something called biosolids. So biosolids are a result of our wastewater treatment processes, and it's kind of the settled, you're a bit more familiar with the process.
Biosolids basically are organics that are coming from our wastewater treatment facilities. So it could be dried solids of like fecal matter or extra greases that are being dumped down our sinks, or it's a bunch of different stuff that is in our wastewater. And what the wastewater treatment facilities do is they're drying out this very rich organic matter and then applying it to our farm fields. So in this process, there are lots of microplastics just from, if you think about how we do our laundering, our wastewater is actually partly our laundry water too. So all the microplastics coming from your clothing are also in the biosolids, which we apply to farm fields. So that can be a source to agriculture, as well as if you think about certain crops like ginseng that use big plastic tarps for shade in their covering, this can also break down a little bit and provide microplastics from agriculture uses.
All right. Hopefully that answered the question. I thought that was pretty good there. This question comes from Michael Young. What were some of the hurdles you encountered when you were doing the field study and trying to gather samples along the river? Thanks. Along the Thames River, sorry.
The Thames River. So I was part of the fish study for my master's while I was at Western. And I think one of the issues we had, one of them was it was COVID while I was conducting my research. So it was a little tricky to get research permits just because it was a slow process. And then the other, I guess, potential slowdown was originally we wanted to sample fish in the lower Thames area just to stay consistent with our sediment study. But what we were finding is the lower Thames area had a lot faster moving waters and deeper than the upper Thames. So when we're trying to sample fish in this area of the river using techniques like backpack electrofishing, which shocks the fish, or seine netting, which is just kind of dragging a net through the river, we can't physically get into the water safely to do our fish collections. So those were some of the hurdles we had when we were trying to get the samples in the Thames River. But generally, if you're sampling sediment, it's a lot easier because you can just kind of scoop it from the shoreline with a shovel, or you can get into the river with something called a ponar grab, which is kind of like a claw that will scoop up sediment. So you don't actually have to get in the river to collect these samples.
Very interesting. All right, and this one comes from an anonymous attendee. We have, what can individuals do to reduce the fibers coming from our clothing?
Yeah, that's a really good question. I think one of the biggest ones, well, I think we're all kind of aware of how many fibers come from our clothing when you're taking out your dryer lint, and you just see the sheer amount of fibers that have come off our clothing items. So I think the biggest one is definitely washing less when we can. Another helpful one is wearing natural fiber clothing like cotton and bamboo. But I think washing less is definitely a really big one. And also they're trying to introduce it into Ontario a little bit more. There's washing machine filters that they're trying to introduce, and those kind of attach to your washing machine and act as a filter for the water, and they're trapping all of those fibers that are coming from the water that's washing your clothing. And I think that's going to be an important one. But in the meantime, it's just making sure that we're doing our laundry in a way that we're taking care of making sure that it's not adding tons of fibers. So yeah.
I think another one we could probably agree on would be to reduce fast fashion. So certain brands that are generating new styles constantly, and then donating those when you could still be using it. I think that also is a big thing with helping to reduce our microplastic fibers.
All right, then we've got a question here from Donna Small. Just wondering what farmers can do to reduce microplastics.
It's a good question. We're definitely more on the aquatic side. There's a few people in our lab that work on the soil stuff, but it's definitely, I think, being conscious of if we're applying biosolids, where the biosolids are coming from, and what's in the biosolids as well. So microplastics, we've been doing studies that have been finding just crazy amounts of microplastics in the biosolids that are being applied. And when there's rainwater events and it's kind of running off the fields, then those microplastics can move across different environments. So I'm thinking just being conscious of sort of what's being applied, and also taking steps to make sure that there's proper drainage and it's not just kind of running off altogether.
Excellent. All right, this question comes from Brian Kimmert. Were mussels and or mollusks studied for accumulations of microplastics?
Yeah, so our lab is very involved with mussels. Colleen actually did a study with mussels in her undergrad with microplastics. I don't know if you wanted to touch on that.
Yeah, so I studied mussels in my undergrad just in the Grand River, just to see if potentially they were ingesting microplastics in the environment, and they were.
Mhm.
And one of our co-supervisors at Guelph is very big on mollusk toxicology testing.
Yeah.
So we do have someone in our lab studying mussels in lab, exposing them to microplastics the way Quinn is.
Yeah.
We had another student in our lab do a field study the way I am with mussels in the Grand River.
Yeah.
And then mollusk wise, I guess you do snail
Yeah.
toxicity testing. So there
Yeah.
is some representation for our mussels and snails.
Yeah. Well,
Cool.
mussels are especially important just because of the sheer volume of water that they're filtering on a daily basis, and they're bringing in those microplastics. So that's a really big topic that we're looking at in the lab, is their potential for accumulation and how many microplastics they can take in, and if they're able to get rid of them.
Mussels also have a special place in our heart just
Mhm.
because they're one of the most imperiled freshwater species. So I imagine that the Lower Thames Conservation also has a wide variety of freshwater mussel species that we're all trying to protect.
Mhm.
Yes, we do. We do.
I'm gonna jump in on that for just a second. We actually, the Lower Thames watershed is tied right now with the Sydenham River for most diversity of freshwater mussels. So it's definitely something that we are trying to protect for
Mhm.
sure.
Yes, near and dear to our hearts, that is.
Yes, they're underloved. I think not many people are very familiar with them, and they're incredible creatures.
I would have to agree with that. Got another question here from S. Tweedle. Have you noted microplastic migration from livestock into private drains, and or have private drains, municipal drains, been sampled for microplastic levels?
So I know of a study that was being conducted at Western that was trying to look at microplastics or even just larger plastic items being washed into what is it? The city street grates.
The storm drains.
Yeah, the storm drains. So I know there's a study being conducted trying to catch what's being rinsed in, but I'm not sure that that study has been published yet. I know stormwater ponds are being studied right now. That's kind of a hot thing,
Yeah.
just to see what's rinsing in because they don't get drained or scraped out, like all the sediment taken out very often. They tend to be a place where lots of plastics are accumulating.
Yeah. Yeah, and I think the municipal drains as well, sampling for microplastic levels, the biggest issue right now is the tire wear particles on the municipal drains. So again, like Colleen mentioned earlier on, your tires are breaking down, they're producing the small plastic particles that we call tire wear particles. But when there's rainfall events, those get washed into the stormwater drains and then eventually either go to the wastewater treatment plants or wherever they're outletted to.
Excellent.
All
All right.
I know.
Those are the questions I see now. We'll give it a couple more minutes if anybody else thinks of anything else. But it does look as though the you guys have put your email addresses down there. So I'm sure if anybody thinks of another question, this has really opened up a lot of, I think, topics for debate for sure. So I'm really thankful for you, the two of you, coming and doing this for us, as well as everybody who participated and showed interest in this webinar. I think that this is just, it's always been a topic that I've always been interested in and thought lots about. And I'm glad to see other people are taking the same interest in it. And I mean, all the little things that we can at least do ourselves and look up research to see what is being done on a larger scale. So I do thank you for this.
Yeah, there's always lots and lots of cool research that's being done, especially across Ontario.
All right, a couple more questions are sneaking in here. So this one's just from an anonymous attendee. Just out of curiosity, have you ever come across a study that investigates biodegradable plastics and their impacts to freshwater bodies?
I read a study recently that was looking at biodegradable plastics. I'm not sure it was a freshwater study or marine study, but they were looking at the toxicity compared to regular plastics. But I don't think that they found any significant differences between the biodegradable plastics. But in terms of their impacts to freshwater bodies, I'm not sure. And I'm also not sure about the regulations between what biodegradable versus compostable really means. So sometimes biodegradable could mean that they're just breaking down further, but they're still staying as microplastics, whereas compostable means that they're actually fully breaking down into organic material, but I don't know, have you come across? I know that it's a very big new area of study, but I think freshwater is also understudied.
Yeah, I don't know if freshwater bodies and bioplastics have a lot of studies. I know sometimes there's questions about bioplastics in that they will break down only under specific conditions. So when you throw a bioplastic in a landfill with everything else, it doesn't tend to break down as intended. So I know this is kind of a caution, and municipalities, I think, need to develop better ways to sort the plastic, but
yeah, in terms of the biodegradable plastics and impacts to freshwater, I think that's definitely going to be coming up a lot more in the literature. Typically, marine bodies of water are more the focus of the first sort of rounds of research, but then freshwater ends up being a little bit understudied, which is sad because our freshwater systems are incredible and important. But yeah, there'll definitely be something that will be coming up more, I'm sure.
Excellent. Well, I'll certainly keep our eyes open for that.
Mhm.
This next question comes from DJ. Where is the research being done on beet juice and potassium issues?
So we did some studies with beet juice and mussels, the Glaucidia, which are the parasitic life stage of the mussels, which seems a little bit confusing for anybody not familiar with the mussels, but they're just the larva of the mussels. But they exposed them to the beet juice, and they were finding that it was more toxic than road salt. It was.
yeah, and this was done in, was it Environment Canada or University of Guelph?
I think it might have been Environment Canada.
Yeah, it's up and coming. And if you're interested, I think there was a news article published. If you just Googled beet juice and road salts, I think it was Ryan Prosser who answered those interview questions.
Yeah, that's our supervisor. But yeah, we did a few different studies in the lab with the beet juice compared to just higher chloride levels, and it didn't seem to be much of a better solution.
Okay, it's good to know. It's a question that has come up in our office quite a bit, is just alternatives to road salt, because I mean, anybody knows, you get behind a salt truck, you can just see the volumes that get put on when there's like a centimeter of snow, not even. So I don't know, like you said, like if there's ever going to be a solution necessarily, but certainly something to look into as a whole, and for municipalities to jump on board with that as well. All right, it looks as though all the questions have been answered. I'll just put it out there one more time for everybody, and I just want to make sure everybody gets heard if they did have some questions.
I think we might be good there, but yes, again, keep an eye on the email addresses on the screen right now if you have specific questions, or if you have questions about the environment or our watershed in general. Again, please feel free to visit our website or social media and get in touch with either myself, Alyssa, or my colleague Sarah here, and we're more than happy to answer anything you need.
Yep, absolutely.
Awesome. Okay, well then, if that's it, I think we are going to wrap this up here. Thank you again, everyone, for attending. You will all be entered in for a chance to win an LTVCA parking pass for the year of 2024. Very exciting. Every time you attend one of our webinars, you get added in another time. If you attend all 4, your entries get doubled. So come back. It's worth it.
All right, we just have somebody saying thanks for a great presentation. So awesome.
Thank you so much.
Awesome. Okay, have a great night, everybody.
Okay, take care.
Bye.